From page 13 of your student guide, we are going to be looking at the following 4 learning intentions. Looking at mRNA in a little bit more detail with regards to its process and transcription. Going to have a look at tRNA and the process or the role it plays in the process of translation. You need to be able to describe the sequence of events that occurs during the process of transcription and explain what is meant by the term RNA splicing to include what is meant by the terms introns and exons. From our last PowerPoint, we identified what is meant by the term phenotype, which is the physical appearance of organisms, and that's determined by the proteins that are produced as a result of gene expression. Gene expression involves the processes of transcription and translation, both of which we're going to go into in a bit more detail in this PowerPoint. We also introduced the 3 different types of RNA to include mRNA, tRNA, and ribosomal RNA. mRNA and tRNA we go into in a bit more detail, looking at their function and how they engage with the processes of transcription and translation. If we think about the information that's present in DNA, it takes the form of a molecular language called the genetic code. That genetic code is represented by the sequence of bases along a DNA strand, which we've come to be familiar with when we've looked at the DNA structure in our last chapter. So what we're going to be focusing on here is the importance of that genetic code being copied in such a way that it is able to translate into the protein that it's designed basically to build. So when we look at that genetic code or those sequence of bases that represents the code word to make a protein, we're focusing on how an individual cell is able to encode that DNA to produce a particular order of amino acids that is going to code for a particular protein that's needed by the individual. The genetic information for a particular protein is carried on a section of DNA in the nucleus. So what we're looking at is, if we look at the DNA content of a nucleus, when we're talking about transcription, we're only talking about one particular gene being transcribed. We don't look at the full chromosome being copied into a molecule of mRNA. So the problem we've got with gene expression is that the genetic information to make the protein is in the nucleus. However, the assembly of amino acids to make the protein is carried out on ribosomes, which are in the cytoplasm. So this is where we see the role of these 2 RNA molecules. A molecule of mRNA, messenger RNA, is formed or transcribed from the appropriate section of the DNA strand, and it carries this information out to the ribosome. So think of the M for messenger. That messenger RNA is carrying that information from the nucleus to the ribosome. It's at the ribosome where the mRNA meets that other RNA called transfer RNA, where we see that genetic information being translated into a protein. DNA possesses 4 different types of bases. Those bases we've been looking at are adenine, thymine, guanine and cytosine. If we look at proteins, however, proteins contain 20 different types of amino acids. That's 20 that we are aware of. So if we look at one base coding for one amino acid, we would actually only be able to code for 4 different amino acids. And as I've said, there are 20. If we take a sequence of 2 bases coding for an amino acid, there's actually only going to be 16 potential amino acids that can be coded for. So this is where we find that if bases are taken in groups of 3, then that actually gives us 64 possible different combinations, and that sufficiently meets the fact that there's 20 different amino acids. So it is now known that each amino acid is coded for by one or more of these 64 possible different triplets of bases. So in an individual's genetic information, what we look at is the triplet of bases, and that triplet of bases will code for a particular amino acid. So on the screen in front of you, you have what's called a codon. So in a messenger RNA molecule, each triplet of bases is called a codon, and each codon corresponds or codes for a specific amino acid. So on the screen in front of you, you can see an example highlighted of what we call a codon or a triplet sequence of bases. And those triplet codes on the mRNA sequence interact with what's called an anticodon on a transfer RNA molecule, which is picked up and is complementary to one particular amino acid. If you have a look along the mRNA code, you'll see there that there are a sequence of different triplet bases, and they all code for a different amino acid. There's a little transcription task here on the screen for you, and I would ask that you take your time over it. It's a good way to learn the coding. It's a good way to recap your base pairing rule, and it's a practice of looking at these grids, because these grids are quite common in past paper questions. So you've to state the amino acid coded for by the codon GAU, state the codon for amino acid TRP, and identify the DNA sequence for the amino acid MET. When you're ready, you can move on to the next slide. Here's another task for you to try, again, just to practice your DNA sequencing, transcribing it into messenger RNA, and then seeing if you can use that table to translate your messenger RNA sequence into an amino acid sequence. So your task is to write down 15 DNA bases. So remember, that's thymine, adenine, guanine, and cytosine. You've then been asked in B to transcribe it into messenger RNA. Please remember that in messenger RNA, there is no thymine, it's replaced by uracil. And then in C, using your messenger RNA sequence, can you translate that into your amino acid sequence using the grid on the screen? Transfer RNA, or tRNA, is found in the cell's cytoplasm. It's composed of a single strand of nucleotides. However, a molecule of transfer RNA has a three-dimensional structure because it's effectively folded back in on itself in such a way, hopefully you can see on the diagram, that there are hydrogen bonds that form between many of its nucleotide bases. So if you take the diagram that's in front of you and stretch it out, take the 5' end, take the 3' end, you would actually be able to stretch that out into a single strand. What's holding it together at each of those loops is the little lines going across the middle is representing a hydrogen bond. Each molecule of transfer RNA has only one particular triplet of bases exposed, and these are called anticodons. This triplet of bases, this anticodon, is complementary to an mRNA codon and corresponds to a specific amino acid carried by the transfer RNA at its attachment site. There are many different types of transfer RNA present in a cell, basically one or more for each type of amino acid. Each transfer RNA picks up its appropriate amino acid at its site of attachment, which is represented at the very top of the diagram there, where you can see the 5' and the 3' bit, where the pink bit is. The amino acid is then carried by the transfer RNA to a ribosome, and it gets added to a growing end of a polypeptide chain. Now you should be able to see the interaction, the complementary base pairing between the anticodon in the tRNA and the codon on the messenger RNA. So this is how we're able to get that message from the DNA to the ribosome in the form of messenger RNA. But the codon in a messenger RNA is corresponding to an anticodon on the transfer RNA. So this is how it all communicates, and we get that transcription and translation from the genetic code into a protein. So we've got a task here again now, looking at the process of translation. So this is using transfer RNA anticodons and seeing what amino acid they represent. You've got a question there about what order of amino acids would be synthesized from the base sequence of DNA shown. Now this is a very common question, starting with DNA, and you have to work your way down through messenger RNA, then to transfer RNA, and then carrying out the process to say which amino acid sequence it would be. Let's work through this one together. There is our base sequence of DNA, and I've just copied that directly from the question. So we've got CGT, TAC, GTG. Now the first thing you've got to do is transcribe that DNA sequence into messenger RNA. So using our base pairing rule, the transcription becomes CGA, AUG, remember there is no thymine on messenger or transfer RNA, and then followed by CAC. What we then need to do is figure out the complementary anticodons that would appear on a transfer RNA molecule. And they would read as CGU, again remember adenine to uracil because it's RNA, UAC, GUG. And the last step is now to figure out what the translation would be. That's our genetic code, that's our triplet of bases as they're read by the ribosome, but what would be the amino acid sequence? So CGU would correspond to alanine, UAC would correspond to methanoine, and GUG would correspond and translate to histidine. So for translation, we see the translation is the process by which a protein is synthesized as a polypeptide chain under the direction of the messenger RNA. So the messenger RNA has attached to the ribosome. The genetic message is being read as a series of triplet bases. So the messenger RNA attaches on and it starts to read each triplet base on the messenger RNA code. This corresponds to a complementary transfer RNA meeting up with the messenger RNA on the ribosome, and each of the amino acids that's brought in sequence are aligned in the correct order. The process of transcription is the synthesis of messenger RNA from a section of DNA. A promoter is a region of DNA in a gene where basically transcription is initiated, transcription begins. RNA polymerase is the enzyme responsible for transcription, as hopefully you can see in the diagram. As RNA polymerase moves along the gene from the promoter, because that's where it all begins, unwinding and opening up the DNA strand occurs because basically the RNA polymerase is breaking the hydrogen bonds between its base pairs. So we're seeing that kind of opening up of the gene by the weak hydrogen bonds being broken. But the role of RNA polymerase, and I love this diagram because it's got wee hands and it's picking up the RNA nucleotides and matching them with the exposed DNA bases. So as a result of the base pairing rule, the messenger RNA gets a nucleotide sequence that's complementary to that initial DNA strand. RNA polymerase can only add nucleotides to the 3' end of the growing mRNA molecule. The molecule basically elongates, so this molecule of mRNA is transcribed as the RNA polymerase moves along the gene. It then reaches, the RNA polymerase will reach what's called a terminator sequence of nucleotides on the DNA strand, and that will be the signal for the mRNA strand to separate from the DNA template and become known as the primary transcript of messenger RNA. The enzyme RNA polymerase moves along the DNA molecule, unwinding that double helix and breaking the hydrogen bonds between the bases. Please remember that for the formation of a messenger RNA molecule, we're only transcribing one gene. We're not transcribing the full chromosome. It's gene at a time. RNA polymerase synthesizes a primary transcript of messenger RNA from RNA nucleotides by complementary base pairing. So as the RNA polymerase moves along the gene, it comes in contact with the DNA bases and it aligns or matches an RNA nucleotide based on the base pairing rule. What we can see in the diagram is a sequence on the DNA. We'll talk about introns and exons in just a wee second. But the molecule that's made as a result of transcription is called the primary transcript. So the primary mRNA transcript is formed by the action of RNA polymerase. Normally, the region of DNA that's transcribed to messenger RNA is about 8,000 nucleotides long, yet only about 1,200 of those nucleotides are actually needed to code for the average protein. So what we see is that long stretches of DNA exist within a single gene, but not all of those parts of the gene actually code for the final protein. There are non-coding regions that are called introns, and then there are coding regions that are called exons. So when we look at the primary transcript of messenger RNA, the primary transcript of messenger RNA contains these exons, which are coding and are needed to make the final protein. But there's also areas called the introns, and they don't code for anything in the final protein. So what we've got to see is a process called RNA splicing take place. So remember, all of this is happening inside the nucleus. We've got the primary transcript now that has been made as a result of the RNA polymerase action. What we see in splicing is that the introns are cut out and removed from the primary transcript of messenger RNA. And then the exons, which are the coding area, are spliced together to form a messenger RNA that has a continuous sequence of nucleotides that will code for the final protein. So the order of exons remains unchanged during the splicing process. The modified messenger RNA, so what that means is the the RNA that has only got exons is called the mature transcript of mRNA. And it's this mature transcript of mRNA that is going to pass out of the nucleus into the cytoplasm, where it then is going to move on to the next stage of protein synthesis. It's basically going to attach onto the ribosome and get translated. So to recap on what I've mentioned in the previous slide, this is obviously just an opportunity for you to take the notes. But RNA splicing forms what we call the mature mRNA transcript. Through the process of mRNA splicing, the introns that are non-coding regions from the transcript are removed, and the exons that are coding regions and are going to go into making up the final protein are joined together to form the mature transcript. In terms of RNA splicing, there is a very, very common core question that gets asked at higher level. So I want to take the opportunity to work through this question with you. We've got a diagram that shows the stages in the synthesis of a polypeptide, so the synthesis of a protein. We've got the chromosomal DNA at the very left-hand side. Stage 1 sees a primary mRNA transcript being produced. Stage 2 sees a mature transcript being produced, which then in stage 3 ends up synthesizing a protein. So you've been asked in part 1 and part 2, explain why the primary mRNA transcript is so much shorter than the chromosomal DNA. And then if you look to stage 2, you've then to explain why the mature mRNA transcript is shorter than the primary mRNA transcript. So at stage 1, what we're seeing is that chromosome, remember, is made up of many genes. When we're looking at synthesizing a protein, the process of transcription only copies one gene. That chromosome is made up of many genes. So the reason why the primary mRNA transcript is so much shorter is because there is only one gene being transcribed. When we look at question 2, explain why the mature transcript is shorter than the primary. Well, what's happened in stage 2 is that process of splicing. So the primary mRNA transcript is made up of introns and exons. But to get to stage 2, in that shorter mature transcript, what we see is that the introns, or those non-coding regions, have been removed through the process of splicing. I would ask you here to evaluate your own learning based on the 4 learning intentions that we've covered during this PowerPoint.